How many classes on how you do Five. Five. Not that with engineers. Yeah, 5 is not bad. So it's 3 credits each.
Yeah.
So like 15 hours. Right. Oh. The problem when the class is not divided like this, it cannot be very heavy. I'm looking at classes from previous year, the 1 hour, 20 minutes, our lunchtime here. Because, you know, after your first 1 hour and a half, you are tired. I don't want to just talk to myself. There's a lot of stuff. So, unfortunately, it wasn't. I will try to find more ways. Problem that I'm also really with you. I will stop. So I will try to get it away. Probably I will try. Let me see this Thursday how it look like. And why I'm asking you to come to the office hour because I want to understand how you're getting the things from the teaching material. Not necessarily that you will ask questions that probably very difficult, you didn't get to that, or it's more about how you get with the material. Are you able to understand? Are you able to navigate the slide? Are you able to go to the reference and check that reference? It makes sense what you see, stuff like that. Because sooner or later, you have to do it on your own in your life. And now you have a course. This course, I'm responsible to deliver for you. So I'm responsible to get you all the material you need. But if you go for an honors project or a master's degree, you would have to read yourself. And I don't want to tell you how many papers are published every day if I publish. You already know that. So you have to funnel kind of what references you read, what are the good ones. And you have to get the information adequate as possible, so as accurate as possible, so that you can move on with your project, research, or whatever. So this is a micro project for you to read the slide and understand it. And if you don't get it, you go to the reference. Read that. Again, it's not always pleasant. That's how it works. Okay, so just for your colleagues who were on Zoom and they didn't get the point from that couple last of slides, I will very quickly go back to the IGF slide. I put them back on track with us. So in this, we are discussing the post-translational modifications and how to play a role in the signaling. The protein IGF is one of the ligands we are discussing here. Once it's available outside of the cell, it has to bind to the receptor. Once it binds to the receptor, now if the situation as we are describing here, the internal part of the receptor is inactive, and it needs that signal from the ligand in order to become active. And I mentioned that when the ligand is present, then the receptor has a conformational change, and that conformational change most probably will cause this inactive protein to become active. So this inactive protein here become visible, and therefore probably now we can phosphorylate it. And now once it's phosphorylated, it becomes active. And it works almost in every signaling that we are discussing, dephosphorylation in this process causes a proliferative effect. So therefore, the more IGF we get, the more activity we get, and in cancer conditions, these induction of the IGF could propagate the cancer much faster. The second example I gave was the neutralizing antibodies, and these neutralizing antibodies could be used as therapeutics, and particularly blocking the binding of the IGF to its receptor. And therefore, you could minimize this signaling pathway and minimize the proliferation of cells. The second example is the usage of these neutralizing antibodies as therapeutic particles that could block the viral infection at multiple levels, either sequestering these viruses outside of the cell, preventing them from binding to the membrane or crawling there, internalization, processing, and the exit. All that could be blocked by neutralizing. I see some chat. Okay, sounds good. So the second example I gave was the glioblastoma protein, and the glioblastoma is a tumor suppressor. As the name says, it suppresses tumors because it keeps the check over the cell division and proliferation. When the signal is received by a growth factor to the receptor, then the phosphorylation of the glioblastoma causes the release of these transcription factors, and it causes the activation of the proliferative step. And therefore, if goes outrageous, it causes the growth of the cells, and this is what happened in the case of glioblastoma. Okay, sorry, I'm repeating this again, but now your colleagues are with us on the same page. Please call these examples. The third example, the neurodegenerative diseases. I listed 4 of them. The neurodegenerative diseases are mostly affected by, in fact, aggregation. So you would see a common criteria in neurodegenerative diseases that there is an accumulation of certain proteins that could cause neurodegeneration. And therefore, if you look at that, thinking about these proteins being present, we could identify 3 or 4 of these proteins that, in fact, get post-translational modifications and the change in their function that could cause the generation of these molecules or these proteins being aggregated. So the first one is the tau protein. I'm pretty sure we heard a lot in 2010, 2011 about the mad cow disease and all that, where these proteins are being aggregated. The process of getting these proteins activated to become aggregated is a simple phosphorylation. Therefore, it causes these neurofibrillary tangles to be generated because the tau protein makes like tandem repeats. And then it becomes very stable. And once it's very stable, it's like an aggregate that the body cannot let the neurons cope with, and they could cause Alzheimer's. Similar thing happens with the TTP43. I gave you what is that in the other slide, which is a processing molecule at this level of protein transcription, messenger RNA translation. If you phosphorylate that protein, it becomes hyperactive, and it leads to the aggregation of those proteins together in the neuron. This would affect the muscle neuron. That's mostly in the ALS protein. The second one on this slide is the syncytin, which causes the Parkinson's disease. It's similar aspect. you have a phosphorylation event, one single phosphorylation event that could trigger the process of transferring this protein from the native molecule to a toxic file. This molecule, when it's generated in different repeats, it causes the aggregation and the formation of this Lewy body. And in principle, that's the major cause of. The third example is the Huntington. And now the phosphorylation event is playing in a different opposite. When you have the phosphorylation, it's a good thing. The dephosphorylation causes it. Okay? So it's not always that phosphorylation causes an activation over. Now you have Huntington with a muted form of Huntington. If it's phosphorylated, it's good. Now the dephosphorylation causes the aggregation. So hypophosphorylated causes it. And I just told you about the DP in the previous slide. So this is another example. Now, I didn't tell you what is the symptoms of those. Probably by the end of the lecture, when we have transcription, translation, metabolics, cell division, communication between the two different types of cells, then I will describe it a bit more this one more time. But that's one example of a post-translational modification that could cause, so far, 6 diseases, which is that 4 neurodegenerative diseases and 2 neuroblastoma and the cancer. Okay. Let's stop in this slide. So normally, I always like to have overview that could be useful for you to sit down and watch and not listen, like don't listen to me. Sometimes it's made by a very professional company with a lot of animation. So I have 4 of these that are listed for you. Anytime you want to review the lecture, I would recommend go first, watch this video, and then go to the slide. It will make a lot of sense for you. Obviously, I don't want to waste a lot of time in the class because you know you will get bored and we just come back from the break. So I need you to be focused for the next 20 minutes because I will lose you around 10:30. I know that. So, okay. So we spoke a lot about proteins, but now let's go back one step. We're gonna go back and make like a tour how the proteins are made, and then almost we will meet back together at the level of post-translation. So now we go back to the genetic material. The proteins come based on a specific DNA sequence, a gene. So simply put, what is a gene? Who can define a gene and who can tell me how many proteins you can get out of it? Don't feel afraid. You have to start interacting. And one day you'll be sitting in a room and you have a professor working with you and then I pose a question. It's not going to kill you if you don't answer questions. Just break this barrier. And your knowledge is very important. Everybody has a knowledge. Experience makes you more knowledgeable sometimes. Don't feel afraid to say what you know. Who can define a gene? Very simple. Yeah.
It's kind of, I guess, a layman's definition, but it's sort of a building block where when you put a bunch of them together, it makes up the transcript that defines who you are. That makes you you.
Yes. But can you find one of them? What is one?
Well, I said it's like a building block, but for that.
Yes. Well, go ahead, I'll come on.
A sequence at the end, like a specific part of the DNA sequence, like the nucleotide.
For what? You got it? Okay, that's all for me. Okay, very simple. It's a stretch or a sequence of the DNA that could be transcribed into messenger RNA, and that could be translated to a protein. And this was the classical definition of the gene, and now it's not correct. Because there are two things you have to remember. I'm not going to the history of genetics. Who discovered DNA? Who found the helical structure of DNA, branches and Craig, all these. So the gene was supposed to be what they described like just a piece of DNA, sequence of DNA that's transcribed into a messenger RNA or a protein. But now we know that there's a lot of genes that will not really generate protein. It could generate microRNA, it could generate long-range RNA. There are many types of RNA that are not transcribed. And there was also an aspect that a gene generates one protein, one gene, one protein. This is not true. Because now we know that one gene can generate more than one protein. The protein could be truncated, and it could be the RNA being spliced. So, the transcription of a gene has to start at certain specific sequence. So we have our genome, only one chromosome, X and Y, and then we have those genes stretched all over the chromosome. Okay? And certain parts of those chromosomes are coding and certain parts are not coding. Right? Let's go very narrow. Let's start from the very basic concept of a gene, and we build it. And exactly as I described the signaling molecules, I will be describing for you how we will use this in the knowledge of diseases. So, after the messenger RNA being transcribed through the transcription process, it will be processed, then it will be translated. So first, we have to know where is this gene, on which chromosome, where it starts, where it ends. So this is number one. Then we would need a machinery that's called transcription machinery to come to certain specific sequences and do the transcription. Then we will end up having a messenger RNA, and that messenger RNA is going to be processed and then translated. Okay. So the building blocks. We discussed this in the previous lecture, and we mentioned that those bonds between the pyrimidine and purines with the ribose or the oxyribose make no difference between RNA and DNA. And of course, when we are talking about these molecules, there is a reason why their stability is different based on the sequences. And this is something we could use, because imagine you want to isolate DNA from a cell. You would like to know the properties of that DNA, because then you would really know what buffer you can use, which pH you can use, so that you can take that DNA out. Otherwise, how would we sequence the DNA? How would we make a gene? How would we clone a gene? We wouldn't know how to do that. So these basic structures are very important to differentiate between RNA and DNA. The video there that I showed you on the YouTube is very nice. Please go and watch it sometime. And I'm covering a little bit of it here. So the 4 nitrogenous bases, A, B, C, G, I think we already have passed through that. And the double bonded triple bond that takes place between the AT and the CG. So one thing that you have to remember the more CG in your gene, probably the more problematic. Because those are very difficult to clone. 3 bonds is more difficult to break than 2 bonds. So GC-rich areas are most of the time problematic for us molecular biologists when it comes to cloning purposes. So a stretch of that DNA, a known sequence could make a part of that chromosome, so this is a gene. And it's most of the time one gene to code to a certain function. And that is made by the protein. And all that is packed into the nucleus. The only difference between RNA and DNA is the uracil being replaced with the thymine. And the helical structure, one is a double strand, other one is single strand. Now, this is very nice in terms of the stability. And you already can tell that RNA is more reactive compared to DNA. Because the DNA is more stable, it's more close. You have 2 structures built together. If we look closer, the pentose in the ribonucleic acid, it has an OH group on the carbon number 2. And that's the only difference you see in the deoxyribose, which is the building block for the DNA. And therefore, now you can think that you could donate this proton on the carbon number 2, and you could react faster. Okay, so how would that translate into like practice? So if I want to extract DNA and RNA separately, now I have a way to do it. It's that simple OH group. But if you change the pH, like you put water at 7, both of them are soluble because they are in our cells and they are functional. Now change the pH, the DNA will lose the activity because now it cannot really react with this guy here. But the RNA is going to be able to still react. And therefore, I wrote it down. The RNA has 2 hydroxyl groups, which it makes more polar than DNA. And therefore, it's more aqueous in conditions where it goes acidic. Because this OH group is going to maintain the RNA able to react with the acid. At that point, if you keep dropping from 7, 5, 4, it's very acidic for the DNA. So the DNA is not going to be any more soluble. And the RNA will be soluble. And that's how we use buffers to separate RNA from DNA. By simply decreasing the pH of the buffer we're using, you automatically separate the DNA in one side that is not in the aqueous phase anymore, while your RNA all go into the aqueous phase. So you see how an OH group would change the process of a whole molecule just by changing a buffer. That's something pretty amazing. So, that being said, we have the 4 nitrogenous bases. And we know that each 3 of those will give us a code for an amino acid. So 1 amino acid needs 3 nitrogenous bases. So you have 4 nitrogenous bases, and you have 3 to create an amino acid or to code for an amino acid. So 4 to the power 3 is that 64, and that's how you get 64 codons in order to make the 21 amino acid. Okay. So I said what is written on the slide. Okay. Good. So now we know we have this sequence which will be transcribed into a messenger RNA. And let me always go back and tell you I'm explaining about one molecule, which is the messenger RNA. Transcription is more complicated than that. So I'm just giving you an example. So you would have here on this side a DNA that is a double strand. And one thing I missed to say on this slide, I want you to remember, is that this first carbon, here, the 5 prime, when it has the 3 phosphates, we call the 5 prime. And as we are going forward, we have another one that already had 3 phosphates. It donated 2 of them. And now we have this phosphodiester bond. So this is one nucleotide, this is another nucleotide, this is the third nucleotide. Obviously, here they are not showing you the nitrogenous bases. They just wrote the base. So you have A, T, C, or whatever at the place of the base. Okay, are you okay with this? This is clear for everybody? Okay, so if I go back there, this 5 prime end on one side of the DNA, it will be ending at a 3 prime end because that last molecule added, it doesn't have a phosphate group, the 5 prime. Okay, they are complementary. And you could see that it's a reverse of complementary. So now you have a strand that is, we call it sense. Yes, we call it sense because we think about the gene going that direction. And we call it antisense. I think this in nature, it doesn't mean. So for us in this slide, we have the sense strand, which will be probably transcribed. Many, many times you will see that the gene is in fact on the antisense strand. So the gene could be this way because you're reading from this 5 prime end antisense. Or the gene could be on the regular like 5 primes strand here. So it could be going this way. But the rule that we should not forget is that the genes are always from prime to the 3 prime. Whether it's sense or antisense, it's from the 5 prime to the 3 prime. Okay, so what do we need in order to transcribe this gene? We need a promoter. So what is a promoter? It's another DNA sequence that is able to be bound by transcription factors, which are protein polymerases that will perform the transcription process. It will come in the next slide. Okay. We have the RNA polymerase. RNA polymerase. So it's generating RNA. But where it's reading the information from? From the DNA. So transcriptional speaking, so it's a DNA RNA polymerase. You will read the information from the DNA and convert it into an RNA molecule. The gene starts at an initiation and ends at a terminal. Each gene has a start and end. So that's great. Now we know that we have a promoter, machinery bind, it copies. When it receives the signal to stop, it terminates. Okay. So we got the generation of that messenger RNA. Now, if we look closer to that messenger RNA that is being generated, it's in yellow. You can see that this machinery comes and sits at the promoter and starts going. It reaches the initiation step. Okay, that's the start of the molecule. It starts copying the DNA into messenger RNA. So we are generating a single messenger RNA molecule. And it's continue reading till it gets there, where the termination stops the transcription process. And now we have a release of this messenger RNA. Now, the RNA polymerase doesn't have a capacity to think what to copy and what not. It will start copying at the initiation, it stops at the termination. We will have this pre-messenger RNA or precursor messenger RNA. It's a very simple mirror image of that DNA. With one simple modification, the timing is a uracil and it's only a single strand. So we copied only either the forward or the reverse strand. Then we will go to the processing, and I will have a slide to describe this, and then we will go to the translation. Okay, step one is okay, everybody? Cool. So this is the transcription initiation. So there are many RNA polymerases. There are 4 of them. I don't want you to memorize what RNA polymerase synthesize what type of gene, because you will forget it tomorrow. Polymerase number 2, and I'm pretty sure you will remember this for life, is the one that transcribes the messenger RNA. Okay, polymerase number 2. So in eukaryotes, that's to be specific, the polymerase, RNA polymerase number 2. So what we are talking about, RNA polymerase number 2 is a protein. So recall what I told you in the beginning of the class, that proteins are playing a role in generation of protein. So the protein is reading the information, the genetic information in the DNA, make it become transcribed to RNA. So this is going to become a protein. That gene, once it's transcribed, it will become a protein after translation, and it will perform the cellular function. So the RNA polymerase binds to the promoter region. So that's what becomes very important, that promoter region. Because the RNA polymerase will come and just start scanning, as if I'm looking, for example, for if I say that the promoter is a sequence of ATCG, AT, and just for one. And for me, it looks like this, water bottle. So as a polymerase, I will be coming here and I'll start scanning and scanning for the sequence. Okay, I'm the promoter. So as a polymerase, I will stick to this sequence. So downstream, this is going to be my gene. So that's my promoter. But where the messenger RNA is going to be starting, it doesn't necessarily mean it's just happened. It tells me that downstream, there will be a gene. The gene could be here, the initiation. But the promoter starts then. There could be a space or sequences between the promoter and the initiation. So as RNA polymerase, I found that. And this is very important for you to know that when the RNA polymerase would bind to the promoter, it needs other transcription factors to support. And that's what you see here, general transcription factors. They are very well known. These transcription factors, did I write it down? Yes. So the transcription factors, these are proteins that control the activity by binding to specific DNA sequences, regulating whether the genes are turned on or off. So again, that protein, so probably the RNA polymerase will need 3 or 4 factors to know where exactly. Okay, so simply put, this RNA polymerase is going to start scanning somewhere here, moving. Transcription factors probably would help. And then the RNA polymerase is going to bind to the promoter region. And your gene is going to be transcribed somewhere here. Not necessarily just at the site of the promoter. It could start somewhere a little bit downstream.
Okay, and the core promoter elements, we know from genetics that there are certain sequences are really common between many promoters. That many genes, when they are transcribed, they start at these promoters. I put one of you, I'm pretty sure you heard about it before, the data box, which is a TA and 2As upstream. So somewhere here, there's a data box. Okay, and there are these B recognition proteins. These are transcription factors. There are a lot of these transcription factors. I'm not going to go into the detail. But in order to generate this complex, which is RNA polymerase protein number 1, transcription factor 1, 2, 3, and 4, whatever. So this complex of proteins now is at the site of the promoter. And it will be transcribing the messenger RNA. Of course, we will need more support. We will need a helicase, for example. You know what's a helicase? Right? The helicase is the enzyme that will unwind your DNA. Because now it's very difficult for this protein to access. But we need to unwind that. And this will continue. The gene will be transcribed. Okay. So some of those is summarized in text here. So you can see that the RNA is start this side being generated from the 5' end to the 3' end. Again, it's always gene 5' to the 3' end. You're generating that messenger RNA, copying the template, and stopping at the site of termination. Okay. So here I put you more text about what is the promoter, how you can find the promoter, how the RNA polymerase finds the promoter, and then how we define a gene functionally now. So now we have a part of the DNA that is the promoter, not part of the gene. Don't get confused between the two. The promoter is not part of that gene. It is the sequence needed for transcription of that. Because we don't code the promoter. We don't translate the part. Okay? That part here. So now you have a depiction of a non-template strand and a template strand. So this means the gene transcription is taking on the lower part of that gene. Then you would have the promoter region. This is normally on the 5' end, we call it upstream. And beyond the termination site, we call it downstream. So upstream is always where you find the promoter. And downstream is always where you find the termination of it. And in between, you have got the 5' to the 3' RNA. Okay, we coded that RNA. We copied that gene sequence. We transferred the thymines into uracil, and we have it as a single-stranded messenger RNA. From the initiation site to the termination site. Okay, so now we have that messenger RNA. Is it going to be functional? Is it going to be all translated to a protein? This is a different story, which brings us to the next step. So when you get that messenger RNA, so the RNA transcript, we call it RNA transcript. And nowadays you hear a lot about transcriptomics. Transcriptomics or transcriptional analysis. We hear a lot about these analysis and these types of studies. So when I say I'm studying the transcriptional regulation in the cell, most probably I'm looking at the transcription of all the transcripts. The transcriptome is all the transcripts that is being transcribed in the cell. This means all the genes being transcribed to messenger RNA, this is the transcriptome. So you can imagine how many thousands or millions of those. Okay, so we generated these transcripts. All the transcripts will make the transcriptome of the cell. And now, if I zoom into this green part, you can see that it already has this pinkish color that is added to illustrate that, yes, it's just the termination site there, but this part of the transcript, most of the time we want to translate it. So let's zoom in this green part down there. Okay. So what happens? Now we get to the next level. We need to translate this into a protein. Of course, in order to translate that to the protein, we will kind of have the same type of circuit. So the ribosomes, in order to translate this into a protein, they need a little stretch of that messenger RNA in order to recruit the machinery, right? And this is why most of the time we have the 5' untranslated region. So this part here is not going to be translated. It's not going to be part of the protein generated from that gene. Then you would need the initiation, the start codon. And you all know what's the start codon, right? What's the start codon? ATG. ATG, which is the methionine. Every protein, remember the first lecture? Every protein starts with an ATG, which is a methionine. It's the only codon that is generating the methionine. So there are some sequences that direct the translation in bacteria. It's called Chandler-Garno sequence. It's the person who found that sequence. And then you have the start codon, which is in the ATG. And that's where the translation starts. Now, the coding part of that gene keeps going till we reach one of the three: UAA, UAG, and UGA. These are the three stop codons. The translation of a messenger RNA to a protein, it keeps going till we get one of the C: UAA, UAG, and UGA. And that's when the translation stops. And therefore, you automatically know that after this stop codon, a part of this green here is not translated. And that's why we call it's an untranslated region on the 3'. So it's 3' untranslated region. Okay, so we have a 5' untranslated region and a 3' untranslated region, and then the coding part of the protein. Okay. Pure genetics, reading sequences, starting, stopping, polymerase, do all that function. How is this translated to a disease? It's very simple. UAA, UAG, UGA, I just give you one example. These are the stop codons, right? Imagine there was a mutation in one piece. So supposedly, the protein of question started at the start codon. It reached that point, and there's a mutation in the UAA, UAG, and UG. Whatever that stop codon. And then the translation will continue, and therefore, you will be translating part of that 3' untranslated gene added to that gene. What does that mean? Every codon you're adding one amino acid on top of that protein, right? So every 6 amino acid, 9 the amino acid. If you added a stretch of more than one amino acid here, you are having a possibility to deactivate your protein. So that's how genetic disease, most of the genetic loss of a stop codon, a mutation within the coding region here, that could change the amino acids in one part of the protein. And there's many causes of that. We will discuss the genetic disease probably in the lecture 5. That's probably after your midterm. Okay. Anybody has a question here? I will start using it 10:30. That was the one. Question, yes.
But the structure will not be individuated, that's.
Oh, that's, yeah, the translation of it, yeah, English. Speaking about genes, DNA level, very good catch. Did you get what your colleague asked? He said, I'm saying to the first column that ATG. And ATG is the DNA coding for that page. I'm just speaking a lot about molecular biology because I do it in the lab. So every time I'm cloning a gene, we have to make sure ATG on the start. Okay. So during the transcription, the steps of the elongation of the transcription before we get to the protein translation, I just added that for you to see how the chemical reaction. So you can see that you have the DNA on one side is being transcribed from the other side here. And you have the 5' end because you have the phosphate groups here. So this is, we already discussed it, but it's just for you visually to look how the reaction takes place. So you have this being, the A is being transcribed to uracil, right? And that is having the 3 phosphate groups, and that's the 5' end of it. And then another molecule comes here, which is the guanine. And now the guanine, once it arrives, it has a 3 phosphates, right? It has to give out 2 phosphates and make a phosphodiester bond that I described before, so that you get the sequence now bind on one side with the phosphodiester bond, and on the other side, you have the triple bond with the cytosine. Now the third molecule comes in. Since this is a T on the DNA side, on the RNA side, it has to be an A. And of course, the same thing. You have the 3 phosphate groups. You are donating 2 out and making a reaction of a phosphodiester bond like this one here between these 2 guys. And that's how the process continues till the end. Now, there's very important thing that's happening. You are getting a lot of phosphates. Every time you're getting a phosphodiester bond like this guy here, you got 2 phosphates out. And phosphates, we're going to use them in the ADP, so that you are generating a lot of energy that is needed for this process by breaking that phosphate out from the DNA, from the RNA molecule. So every time you're having this phosphodiester bond, you get release of the phosphates. This is a generation, and this is the source of the ADP. Okay? And then we go for the RNA processing. Okay, so we got the messenger RNA process. There is a possibility that we could generate a messenger RNA and it becomes less stable, and RNAs are less stable in general. If you take DNA and you leave it at the bench, and you know that now we can get the DNA from dinosaurs. It can stay for years and can be stable. In the lab, we leave it at the bench, we forget it, it doesn't matter. DNA is very stable. If it has the right buffer, it is stable. But RNA, you leave it at the bench overnight, it's dead. It's very sensitive to the temperature. And therefore, this gives you a snapshot why within the cell, because the cell is mostly water and proteins and some acids and all the components we described, right? Now, it gives you a point that when you generate a messenger RNA, you need to take care of it. You have to make it more stable. So you have to process it. And that's what we call messenger RNA process. So you transcribe a messenger RNA, and then you have to do certain aspects to that. Now, one thing that is always happening is the 5' capping. There are some exceptions though. So you add a cap at the 5' end of that messenger RNA. I think it's there. Yes. So in fact, it's this chemical compound that you added that blocks any reaction to take place on the 5' end of messenger RNA. So you add it at the 5' end here. Then you add a lot of the poly A, which means a stretch of A. You simply add them at the end of the messenger RNA. And sometimes the messenger RNA is cleaved. So it's generated, and then there's enzyme that will cleave that messenger RNA. And the fourth and the most important for you is going to be the splicing. So cleavage of messenger RNA could generate two different types of molecules. So imagine you have this molecule here and that molecule there. The sequence from the 5' end to the 3' end in this molecule, before cleavage, it would be there. So the messenger RNA is different. Now, when you cleave, you generate this guy. So you generated two different messenger RNA from the same molecule. The same thing when you are speaking about splicing, which is in this plot. So the splicing, now we are talking about two different sequences on the messenger RNA. One of them is the exon and one of them is the intron. The exons, these are the stretches that are translated, and the introns, these are the non-translated. Okay, so as I said, the RNA polymerase wouldn't recognize what is an exon, what's an intron, because this is part of the gene. So it simply starts reading from there and copies. So it copies the exons, exon 1, intron 1, exon 2, intron 2, exon 3, intron 3. It found the termination, it stops there. So now we have the pre-messenger RNA that has exons and introns. Okay. So why this is important? Because now you can have a diversity from this pre-messenger RNA. You can generate more than one protein. It's very simple. You have here 3 exons. You can take exon 1, 2, and 3, and that will give you one protein. You could take exon 1 and 3, and that gives you another protein. You can take exon 1 and 2, and that's another protein. And therefore, the RNA splicing is another level of producing more than one protein from the same gene. And that's why I told you the definition of a gene has changed. It's not anymore one gene, it's one protein. Could be one gene, more than one. Okay, so here they give you 2 examples. They take exon 1, 2, and 3, and that is gonna excise introns 2 and 3 out. And this is gonna give you protein number 1. And on the other side, the splicing resulted by exon 1 and 3 only. So you didn't have exon number 2. And of course, this will be a different sequence and a different protein. Remember all the time, splicing takes place in the nucleus, in eukaryotic cells. Of course, prokaryotes, they don't have any. So transcription and messenger RNA processing, and this is all the process that I described from A to Z. This is all summarized here from these pieces. So promoter region, exons are described, initiation, start and initiation and termination, messenger RNA processing, cleavage, polyadenylation, and then RNA splicing. And therefore, now you have from one gene multiple proteins. Then we would like now to translate that protein, that messenger RNA into a protein. So we need the recruitment of many other proteins. Ribosomes are the most important. I'm not going to describe ribosomes for you. I'm pretty sure you know. 2 subunits. And these 2 subunits has to bind to that messenger RNA, scan and start translating. Of course, we will need support from other RNA molecules, and that's why I'm telling you now, not always a gene produces a protein. Sometimes it produces messenger RNA that is not transcribed. And these we call them different types of RNA. So they are classified in the second block. Free messenger RNA that never been transcribed. So this is one of them. Small nuclear RNA, microRNA, single interfering RNA, and now we have long encoded RNA. So the list is getting bigger and bigger. So there's many RNAs that are not transcribed into a gene. But for the translation process, we will need the messenger RNA, the ribosomal RNA, and the transfer. So these are described in the next slide. But before going to that, you all heard about the vaccine that you got, right? The controversial vaccine. Many people make a lot of trouble about, oh, they are changing our genomes. I'm not stating anything about that. I'm just saying people tell me. So, how did we use the knowledge of having this to make a vaccine? So, you know, COVID-19 hits our lungs. It causes disease in terms of our respiration, and many other people die because of that. Now, in order to come with a vaccine, you have to synthesize something that captures that virus. So how did they think about it and how did they make it? So here's one application. So what do we need to know? We have to find a stretch of amino acid or peptide on the surface of the COVID-19 that is very specific to COVID-19 only. It doesn't capture other types of viruses. So that is mainly done bioinformatics. But if you take like now 10,000 proteins and throw them on R, the R language and algorithms, then could you please pick one out of these 10,000 that is specific only to that part? It comes back, it tells you leucine, arginine, glycine. So these 5 amino acids are very specific to the virus. Okay, great. Once I get that information, I can go back and create the DNA. But before I get to that DNA, I can process it as a messenger. Right? Because I know the amino acid sequence, I could create the messenger RNA of that, and then I could create the DNA. The very simple reverse sequencing, right? And that's what they exactly did. So they found that spike protein that is specific to COVID-19. And then they reverse translated. So now we know that the sequence, we went back. What is the messenger RNA that corresponds to this? And then we generate that messenger RNA in the lab. So if we give this messenger RNA to the cell is going to transcribe the protein. Right? Because the messenger RNA is exogenous. We are adding it to the cell. Once it's inside the cytoplasm, it's a very functional, fully mature messenger RNA. Once you add it to the cell, it's going to be transcribed. So it will generate a spike protein inside your cell. And now that spike protein is not self, is not from our body. So our immune system is going to detect it. So these spikes are detected by our immune system. And then our immune system is going to generate antibodies against that spike. And those antibodies, of course, they will be in the memory organ. So therefore, you take that stretch, make the messenger RNA, add it to the cells, cells generate antibodies, you get immunity. You get infected, then the antibodies are already there. They will directly attack that virus. So this is an application that falls in between transcription and translation, and how we could use that to generate a vaccine that millions of people got it. With all the questions that came, that, oh, this messenger RNA probably is stable, is gonna be integrated into our genome, it's gonna change, it's gonna kill some people, I don't know. So, but that's the strategy that was followed. Okay, so we got this machinery of translation. We have the ribosome, the transfer RNA, we have the messenger RNA in association altogether. It will start translating that mature messenger RNA into the peptide. And the way it's going to be doing it, we have this specialized transfer. That on one side, it's an RNA molecule. And on the other side, it's a protein, an amino acid carrier. So it has, for each codon, an anti-codon that corresponds to an amino acid. So as it reads through the messenger RNA, whenever it hits the AUG, we have to have a transfer RNA that carries AUG and matches it to the messenger RNA. And then the process continues. Now the second one comes and there will be the peptide formed. And that's how the peptide being growing and the protein being generated. And at the same time, you are reading through 3 codons each time. That's the elongation process of the translation. Till we reach the end, UAA, UAG, and UGA. Once you get to one of these 3, then the translation process will stop. Okay? There's a lot of this information, I repeated many times, they are written there. Please go through the material. If there's something still missing or a process that is not really clear, I will repeat it in the next time. One thing you have to take into consideration, and probably you will see it in the exams, that like if you look at serine, you will see that UCU, UCC, UCA, and UCG, all of them will produce serine. And you could tell that that last part of the column is the most important part. Because the U and C are common between the 4, and the only difference is that last one. And the 4, if you are thinking about genetic manipulation where you want to remove a serine, you have to think very well about what DNA changes you have to do in order to remove it. Because now, if you change anything within the 2, one of them, it doesn't change much because that's common, right? So it will not be very wise that you change 1, 2, 3, 4 times at the genetic level, at the DNA level. It would be very smart to change one of these, the U or the C. And then you get rid of the serine. You make it glycine or whatever. So it's important for you to know that manipulation at the genetic level and the mutations that we see mostly fall within these two. Okay. So here's an example, the UCC as a messenger RNA, it generates the serine, and therefore the transfer RNA has to carry the anticodon. Be careful here. The transfer RNA is carrying the anticodon in red here, which is AGG. Okay. So translation takes place. Now we have peptides. We generated the protein. The process is terminated. And the amino acid sequence as a simple sequence of multiple amino acids as peptides has been generated. So now we're going back all the way to the beginning of this lecture, where we have a sequence of amino acids that dictate how the protein will fold based on the interaction. Okay, but now we still have to modify these proteins a little bit. Remember the changes that I showed you, the post-translational modification, the folding from the primary, secondary, and tertiary structure of the protein. Now we have to do that. And this happens in the endoplasmic reticulum. Most of these modifications. Of course, some of these protein has to be specialized more. They have to go to Golgi, or they have to go to the membrane. They have to go to the nucleus. I'm not going to start adding all these processes. I want to focus today just on the process of adding the secondary post-translational modification and the folding of the protein. And when it comes to discuss the metabolic proteins, we'll come back to the smooth endoplasmic reticulum. So the difference between the two is that rough endoplasmic reticulum has ribosomes that is attached to the membranes. And therefore, when you're generating a protein, you directly inserting it within the plasma, so it can be processed. The smooth endoplasmic reticulum has other aspects. We will discuss it later. But now we have that protein, we will fold it, we might refold it. And it's very easy to refold it. Simple changes in the disulfide bridges could make the protein, instead of being that, it could be that. We will add some carbohydrates on top, like the glycosylation process. We could assemble more than one peptide together. So we have like two peptides. We make subunits, so the hemoglobin we're talking about in the first slides. And we could package those and ship them somewhere. Like growth factor are being released outside of the cell. And that's how they will bind to a receptor and activate other cells. So protein disulfide isomer. I want you to remember this thing. Protein disulfide isomers. This scheme is showing you that some proteins are being generated at the rough endoplasmic reticulum. The protein as a peptide is being released within the endoplasmic reticulum. And then there's this PDI, protein disulfide isomerase, sitting there and looking for the processing of that protein, making that protein more stable. And they give you just a quick example here. There were disulfide bridges here between amino acid 3 and 4 and amino acid 1 and 2. And therefore, you could see that this protein is not really folding, right? So, but it's more molecularly stable if the peptides make disulfide bridges between amino acids 2 and 3 and 1 and 4. And therefore, this enzyme make the rearrangement so that this protein is more stable. So this is the type of modification that takes place in the endoplasmic reticulum. Last step, Okay, we generated that protein. It went from the ribosome. Now it's being modified. We added all the post-translation modification we need that we described in the beginning: phosphorylation, hydroxylation, acetylation, whatever, name it. With all that, it's still under check. It's still under check. Did it fold correctly? If the folding takes place correctly and it passes by the control, all the quality control steps, then the protein is going to be shipped to the next level, which is the Golgi apparatus or function sometimes. Say one of those steps that I described from the translation process all the way to the rough endoplasmic reticulum and the post-translation modifications was incorrect, then the protein will not fold correctly. And therefore, with all the effort that we put and I described for you, this protein has to be degraded. So it has to be ubiquitinated then and sent to the proteasome. Now, this is not that simple because this guy here could cause stress, cause a lot of stress, and I will describe some in the next lecture, which is called the ER stress. Okay, so we still have half an hour. I will give you like 5 to 10 minutes break, and I will come back for a couple of slides. I'm sorry, we'll have a lecture.
To this, it's. Oh, you said so on actually. Someone's going to come time and then the. Yes. Also, yeah, it doesn't always. That's what they.
Try. Sometimes we could tell that probably are producing like so switching on. Imagine that we have stress that causes this protein here on the and it's so sweet. So you don't have to move in the eye enough. So some of them will some and that's some of them will be going to the next step. Some of them they are not really going correct. But that's one thing.
So let's say this one, where I'm like, this one be selected as expert. Because it's not mobile.
That's mainly all the processing.
So does this one eventually?
Yes.
Okay, so like it's right here. There's like, well, okay. My brain is like,
I do have a reference for that paper. I put the figure to which I do the figure there. But if you go to that article to see this one, it describes in detail this. You don't want to be very.
So it doesn't happen all the time? Or is that?
That's not sounds.
It's the purpose of the English was to get rid of this piece of them off.
Yeah.
Last one.
So the capital 148 every and some proteins that happen or something discovered is not We are and it's related to the size of the and to concentrations. Sometimes you're generating.
You're right. I'm not.
That's actually or one still basis.
Yeah, it's not good.
And your half-life,
That's
so you don't,
Right.
so you don't fully have it.
Okay, seven, that's true.
Some others, like the regular group, and there's one joke about housekeeping, they know. So.
And things like that. So it's that because the mRNA created something that aggravated that.
So there were. Happening. One of them was the macrophages in the pericardium are so sensitive to the messenger RNA generated or translated, and therefore it also activates independent of the protein itself. And the macrophages, when they get activated in that region, is the name called activation of the pericardial area. That was transition after 15 days. They get educated and they process that changes of their own and they will struggle. Oh no, it's not that one response that we have to continue. Then the process goes to help the whole reason.
Just for this information. Say that very excited. People and that's why they're like saying. What is the we're doing in the lab? Like, what is it actually like that? Obviously, it's not nice. Yeah.
Now, and what it is. I didn't get that.
This
You can now,
Yeah, I know, I make chicken.
origin of carbon, hydrogen, oxygen.
Oh, so I mean, like, I'm so scared. I know it's that like.
It's close enough to go get the water. There's no proof.
Yeah.
Some people that it is possible. And from our experience on the RNA in general itself, I don't think so. Today you are wrong. We don't know the messenger RNA. To get to reverse and then insert because that requires 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 reverse transcription of the messenger RNA DNA of the strand.
That's about.
It requires certain beginning of the end, so I don't know how to see that.
It's like. Because like I said, until these I'm finding right now, so this happens. Well, I there's a this stuff. It's like they're paying.
I'm missing my part.
It's like the utilities. So.
This part is
Sorry. If it weren't.
One of the people possible, right? Number 5 is the one that have the possible. Now, the first one if you try to have it. So that's why you.
Yeah.
It doesn't move this one. Okay, and now. If you do that, and now we're going to the next one. The next thing you try to look add this of base. What is the history? You're making something. Here's the thing, I'll make sure guys.
No, makes sense.
So that's one.
I want to know how much.
So. This is the process of.
Yeah.
Has nothing to do with.
Yeah.
Which makes it.
Yeah, yeah.
The four simple sided RNA and simple sided DNA same.
Yeah.
Which takes place at carbon number 3 and phosphating group, and then carbon number 3 and carbon number 5. Then again, carbon number 3, the phosphating group is like that.
Just there. That's so cool.
And that's what you see there.
Right. Well, yeah, there's so much right. It's so straight. Yeah.
And
Yeah.
okay, okay, so let's see what you have still. Okay, disease. First time we hear about disease in this lecture, which is the molecular mechanism of disease. So now, after describing the molecular aspects of itself, molecular aspects of DNA, RNA, protein synthesis, you could tell easily any modification that is outrageous, I would say, out of cell has to be based on the molecular aspects, either at the DNA level or the RNA level or at the protein. So we can define that disease as a disturbance from homeostatic events in a cell. So normally, if a cell is performing a function in a steady state, that all molecules are there, metabolic activity is there. So this is what we call the homeostasis. So the cell is able to perform the function. If it's dividing, it's performing that function. If it's transporting something, it's transporting. If it's a neuron, it's transporting, for example, the senses. If it's a muscle, it's carrying us. All these basics are called homeostasis. Now, not every time a cell goes to a stress, it means the cell has to really die. Our cells are made to adapt. The first thing they do is adapt. And the disease, in multiple cases, is probably because the cells adapt to a level that is not anymore functional. I will give you a few examples. So, when we have a dysfunction of the process, this means the cell has reached a condition where it has to react to stress or a situation. So it shifted from homeostasis into adaptation, and it could not reverse back. Once it could not reverse back, it goes to a disease state. It causes it. Okay? Many of the times I will be describing this for you. So we'll be talking about homeostasis of how the cell divide, adaptation, and then this adaptation went wrong, and then it causes the disease. A very quick example we discussed in the beginning of the class: a cell is dividing normally. It has growth factors that is forcing it to go more reactive, like IGF. It adapts. It continues for long on that adaptation, it becomes cancerous. Okay, a cell is having a lesion, it becomes metastatic, meaning a cancer can move from one side of the body to another side by using it. Right? So it adapts and then goes really into a disease. So here, this is one example, atrophy. Atrophy is a process where the cells become smaller and they die. And then if you look at the grooves of this brain, you can see that the grooves are bigger. That's because there is a loss of those cells. The cells become smaller, 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 and then die. So the cellular changes and the molecular changes that cause a cell to adapt would cause these cells to kind of go to an adaptation state that could affect the tissue. And that collectively is called disease. That's what you are seeing here. Some reason at the molecular level causing a neurodegeneration. You can go back all the way to a phosphorylation event in a tau protein that causes the cell to adapt, to have aggregates. Now I cannot cope with the aggregates. I'm dying. You're dying, you're losing the neuron. So you can go back all the way to that simple post-translational modification event that causes the cell to react and then adapt and then probably die and then cause the disease. So that's like morphologically how we can trace it. And that's one of the examples. So the cellular adaptation has to take place from a normal cell into an adapted cell. And here, the cell is not injured yet. When the injury takes place, there is no way to come back. This injury is irreversible. You want to get rid of that. The best decision to take in the body is to get rid of it when it's irreversible adaptation. Now, if the injury is very mild and the cell could cope with that, then this is irreversible. Let's take one example. Pregnancy. Pregnancy on a woman will increase the pressure on the heart because the volume of the blood and liquids that pregnant women have is increasing the pressure over the heart, right? So the heart has to cope with two types of circulation. One should be embryo, and one because the body is becoming bigger and the volume of the body. So the heart has to adapt by becoming increased. So there is a transitional hypertrophy, which means the heart cells become a little bit bigger to cope with that volume and the pressure that we. After delivery, the body goes back to a normal situation. So that's irreversible. Not an injury, it's a physiological process. So the cell adapted, and then when there is no more reason for that adaptation, it goes back to normal. So I think this is the next slide. Yes. So how do the cells adapt? So there are 5 different types that are listed for you here. The cell might decrease in size, increase in size, increase in number. They could replace, one cell could be replaced by another cell. So what does it mean? Replace one cell in a mature condition by another cell. A mature cell in the pancreas can secrete insulin. An immature cell cannot secrete. If you speak at pancreatic cancer, those mature cells will generate insulin. If the cancer takes place, there will be no insulin generation. So you are replacing one cell by another. Yes, they are very similar, but one is doing the function, one is not. And the dysplasia, which is like a growth that is taking place at different at a side that's not supposed to. So it's an atypical hypertrophy. So I gave you the example of the hypertrophy, which is the growth of the heart cells. Normally, probably that's the size of a cell. In pregnant woman, probably it becomes that much, but that's reversible. So if we look on this one here, this is a condition where there is a disease. And that disease, it causes heart failure because those cells, look how big hypertrophy is the cardiomyocyte here. compared to a normal one. Yeah. Now, at this point, the hypertrophic response is adaptive to that limit that the cardiomyocyte cannot take it anymore. They can't become bigger. This is the maximum. They start dying the heart fails. Okay? I will stop this. Okay, guys. Please come to the office hours if you have questions. Friday will be update on the assignment, survey, and probably on the sums.
Thank you.
You're welcome. Have any questions?
Yeah.